Right, it's time to get started. A warm welcome from the Bioprotocol webinars team. I am Antara Sengupta, and I'm delighted to have you all join us for our webinar series, New Tools of the Trade in Life Sciences, Technologies that Enable Breakthroughs. As many of you know, Bioprotocol is an open access, peer-reviewed online journal dedicated to making life science research more transparent and reproducible. We thank you all for joining us today and being part of this mission. We elated that we can host this webinar today on a topic that has been strongly suggested by the audience community itself. In today's session, we will explore the latest advancements in a powerful imaging technique that enables researchers to visualize biological structures at the nanoscale using conventional microscopes. We will take a deep dive into next generation expansion microscopy, 20-fold magnification in one step. To take us through this exciting technology, we are happy to welcome Shiwei Wang as our speaker today, who will be sharing his insights and expertise on this subject. Today's discussion and Q&A will be moderated by our own Bioprotocol Webinars program lead, Dr. Renee Weiss.
Thank you, Antara. Okay, it is my distinct pleasure to introduce today's speaker, Shiwei Wang, who is a PhD candidate in chemical biology at the Massachusetts Institute of Technology, or MIT, where he develops innovative tools for neuroscience and glycobiology under the joint mentorship of Professors Laura Kiesling and Ed Boyden. His research focuses on developing accessible super-resolution imaging toolkits and visualizing complex carbohydrates such as heparan sulfates in the brain. Shiwei holds a BA with honors in integrated science and chemistry from Northwestern University, where his undergrad research in Professor Will Dichtel's group explored supramolecular self-assembly in macrocycles. He later worked with Professor Yu Yang to study activity-dependent gene programs in the adult mouse cerebellum. His contribution to science and academia have earned him multiple awards, including the Broad Ignite Award, the Whedon Alzheimer Graduate Fellowship, the MIT Presidential Fellowship, the Phi Beta Kappa Honors, and the Best Poster Award at the Chicago Organic Symposium. We are very excited to have you here, Shiwei, today to present your work, and the floor is all yours.
Thank you so much. So first off, I want to thank Antara and Renee for having me here. It's a pleasure to be here and speak to you all about our latest protocol named single-shot 20-fold expansion microscopy. And I want to start with an overview of expansion microscopy. So it's a technology that's first developed by Fei Chen and Paul Tilburg in the Boyden Lab in 2015. And the idea is you can embed a biological sample within a hydrogel that's swallowable. So when the hydrogel physically expands, the biological sample will also magnify physically with it. And also bringing all the molecules apart from each other. So now it's easier to distinguish them using conventional microscopes like a spinning disk confocal microscope. And we are very excited to see so many people in the world already using this technology to advance their science without the need of more advanced equipments or training. So here I have a video of how this technology works in action. So here is a mouse slice that's being processed by the original protein retention expansion microscopy protocol. And you can see once the slice is softened and placed in water, the slice will physically expand isotropically, enabling super resolution imaging with conventional microscopes. And when we looked at the development expansion, as the Renee mentioned, like it has been kind of explosion of different protocols in the world. So here is only a subset of things that have been published. But in general, there are usually like two categories of protocols. One is kind of similar to the original protocol where you have a single round of expansion. And the picture shown here is from the ProEXM paper in 2016, where we first showed that you can perform conventional antibody staining in a cell culture and visualize it after expansion. Alternatively, you can also visualize endogenous fluorescent proteins such as GFP and visualize them with antibody after expansion. And here's also the first time where it's proposed that it's possible to perform antibody staining after expansion, although with this protocol, you will have limited success, and later protocols further improved on that. But in general, these technologies, it's easy to do, but the expansion factor has been limited to around tenfold. And this is largely due to the limitation of the polymer, where if you expand furthermore, the polymer will become weak and the protocol is no longer usable. So to reach higher resolutions, whereas the one-shot protocols with 4 to 10-fold expansion factor, you can reach around 70 nanometer to around 40 nanometer resolution on conventional microscopes. We can use iterative expansion protocols to reach higher resolution, such as 20 nanometer on a conventional spinning disk confocal. We first published the iterative expansion microscopy in 2017, where the idea is you can expand the sample once and embed in a second gel and degrade the first gel and then expand it again. So in that way, they can reach 20-fold expansion within 2 rounds of expansion, and that achieves around 20 nanometer resolution. And more recently, our lab published expansion revealing, which the improvement is that you no longer need to degrade the first gel, which solves a lot of the logistical problems and makes the protocol more simpler, where you can expand 4-fold and expand again to reach around 20-fold expansion and therefore 20 nanometer resolution. However, when we looked at the utilization of the expansion protocols by different labs around the world, we saw that most people end up using one of these one-shot protocols, mostly because they're accessible and there's less workload related to these protocols. Although a lot of people ask our lab, like, how can we reach higher resolutions? Because a lot of the interesting biology happen within the 20 nanometer to 40 nanometer range. So our motivation for this project is to see if we can develop a protocol where it has the simplicity of a single shot expansion protocol, but can have comparable performances to the iterative expansion protocols. And what caught our attention was this chemistry paper that described a novel polymer that is super absorbent, which means it can expand a lot. So here's the original paper that reported this gel can expand tenfold easily. And it's also mechanically robust, meaning that they can stretch these at very extreme degree and the polymer will remain consistent. So that's very promising to us that this particular polymer can be promising to develop the next generation gel system for expansion. And this polymer has been used in a previous expansion protocol called the X10 protocol from the Rizzoli lab. But when we looked at the paper, they didn't really optimize the polymer part. And then we think that we could push this further by optimizing the polymer composition. So what's so different about this gel? So the main difference is in the conventional acrylamide acrylate gel, which is used by almost all the one-shot expansion protocols, we add the sodium acrylate, which is what provides the ionic pressure for the gel to expand. We add sodium acrylamide, we add acrylamide, which is a key component of the gel network. But at the same time, we add this bis-acrylamide that has 2 reactive moieties that can act as a cross-linker between different polymer strands. And that's what holds the gel together. And to reach higher expansion factor, people reduce this cross-linker to allow the gel to expand more. But if you reduce it too much, then the gel will become weak. And that's why the gel is limited to around tenfold. However, for this gel, which we call the DMA gel, it uses sodium acrylate for the ionic pressure. But at the same time, they use this dimethyl acrylamide, which is different than the conventional gel, and they don't use the bis acrylamide for cross-linking. And that's because the dimethyl acrylamide can go through a process called hydrogen extraction, therefore can self-branch to form connections between polymer networks. And that turns out to be very helpful to achieve this very exceptional mechanical strength. So what we started doing is we start to modify this gel network to make it more powerful. So we increase the monomer concentration, basically makes the gel denser. And at the same time, we also increase the sodium acrylate to DMAA ratio to increase the ionic pressure for the gel to expand more. And very quickly, we can reach expansion factor beyond tenfold. However, we reached a problem is that if we simply make this gel over and over again, it's not very reproducible. And that made sense to us because this process of branching is actually a radical dependent process. And if you have oxygen in the gel, the oxygen will quench the radicals and severely change the cross-linking density. So we incorporated different oxygen control method that is new to this expansion protocol, but that allows the protocol to be highly reproducible. And with this protocol, you only need to go through one round of gelation, softening of the sample, and doing immunostaining and expand up to 20-fold. So I want to talk a bit more about the oxygen control part, and I will talk about this more in detail in the protocol portion of this talk. But the two main improvement is instead of typically people using a needle to blow nitrogen into a solution to remove oxygen, here we use a tool that's more familiar to chemists. It's called a gas dispersion bubbler. It's basically a glass sponge that breaks a flow of nitrogen into little bubbles to replace oxygen from a solution very rapidly and efficiently. So with this, we can achieve very good oxygen removal within a single minute. And the other improvement that we use is we incorporate this glove bag. It's basically a plastic bag with two gloves that you can hook up to a nitrogen source. And that allows you to maintain an oxygen-controlled environment with very low cost for a short period of time, enough to construct a gelation chamber to ensure that oxygen doesn't get in, which would reduce the reproducibility of the protocol. So with the oxygen control, we can reliably achieve the same expansion factors under the same reaction conditions. And what we realized very quickly is that because of this gel cross-link itself over time, the expansion factor is actually time dependent. So if you have the same gelation solution, you put into different aliquots, you allow them to gel for different time, and you cut out the same size and let them expand. Different gelation time actually results in different expansion factor. So if you let the gelation to go on longer, you will see a reduced gelation factor, which suggests that by controlling the gelation time, we can have pretty fine control over how much expansion factor we want to achieve. So with these discoveries, we developed a finalized protocol, which we tested with kind of like the biological nanostructures that has been visualized previously with the iterative expansion protocols. So the first thing we looked at is microtubule in cell culture. So this was first visualized with Storm DNA paint and later with the first iterative expansion microscopy paper in 2017. And essentially, you have this microtubule that's stained with primary and secondary antibodies in a cell. And when you visualize the secondary antibody, with high enough resolution, you will be able to see the lumen that's kind of like within this like wall of secondary antibodies. And with 20xm, we're able to see very clearly this gap between the two layers of secondary antibodies, both in microtubules that's parallel to the imaging plane, as well as the microtubule that's perpendicular to the imaging plane. And we did quantification of the distance between the two layers of the lumen, and we saw reliably that this distance is around 62 nanometers, both for the cross-section this way and the cross-section this way. And we measured around 100 microtubule images, and we saw very consistent distance between the two layers. Then this is consistent with the previous Storm DNA paint as well as IEXM protocols. We then further quantified the resolution of these images using Fourier ring correlation analysis. It's essentially an analysis that measures local resolution around, it's basically once you are within a certain distance can you still distinguish signal from noise as a measure for resolution? And we consistently observed a resolution that's below 20 nanometers with a mean of around 80 nanometers. We then quantified whether this protocol introduced distortion to the biological sample. So how we did this is we imaged the same region of a Thy1 YFP mouse brain tissue. So certain neurons in this brain tissue is filled with YFP. And we image the same region before and after expansion and compare them. And we observed low distortion that's comparable to all the previous expansion protocols. And finally, we also apply this protocol to mouse brain and visualized RIM1, 2, and PSD95, which are scaffold proteins for the active zone and the postsynaptic density of one synapse, respectively. And this has been visualized with STORM previously, as well as the expansion revealing protocol. That's the other iterative expansion protocol. So with 20XM, we're able to visualize both RIM1, 2, and PSD95 and visualize the separation of these two signals and able to see the distance in between, which is the kind of the width of the synaptic cleft. And even beyond that, we're able to start to visualize the heterogeneity within these ion channels, within these scaffold proteins along the synapse. So this has been discovered before. It's called little nanodomains. So these proteins, they actually cluster with each other at the nanoscale. And we're able to detect this heterogeneity by performing, this is called autocorrelation analysis, which looks at how clustered the signal is. And for RIM1, 2, we observed that they form clusters that's with a radius of less than 50 nanometers. And with PS95, we observed that they form clusters with radius less than 70 nanometers. And furthermore, we performed cross-correlation analysis, which looks at how closely does these clusters align with each other, and observe that both of these clusters align within 25 nanometer of each other. And from the neuroscience perspective, this is very significant because this suggests that these clusters in the biological sample are closely controlled to align with each other with high precision so that the distance a neurotransmitter has to travel is minimized. And that's important for the synapse efficiency. And all of these are consistent with the previous reports of these structures. And finally, we also visualized nuclear pore complex in the cell culture. And these are 8-fold structures around 100 nanometer, 110 nanometer in diameter on the surface of the nucleus. And we can observe these ring structures with 20XM, and we quantify the radius, how many dots we observe per ring, as well as the distance between each corner. That's consistent with previous known cryo-EM structure. And finally, we start to test different potential applications of this technology. So the image on the left, this is we imaged is in cell culture. TOM20 is a protein on the outer mitochondria membrane, and we're able to use 20XM to visualize good mitochondria hollow structures inside the cell. And we also use 20XM to expand kidney and spleen. And here we performed pan protein staining using NHS fluorophore, and we're able to see the cellular architecture within these samples as well. So in summary, for the protocol of 20XM, we have validated cultured human and mouse cells, mouse brain, and mouse spleen or kidney in our lab. And I saw that a lot of questions were posted about expanding into different cell types and tissues. And when we develop this technology, our hope is that as long as that tissue or cell type has been expanded before with different expansion protocols, this gel is feasible to replace the low expansion factor gel with this gel to achieve higher expansion factors. And the two main considerations in optimizing for different tissues, one is that whether there's enough time for diffusion of the gelation solution into the sample. So if you have really thick sample, or if you have sample that has like a very hard core that might create difficulty for the gel generation solution to diffuse into the sample within the time frame that's enough for the generation solution to expand, to diffuse into, say, like a 50 micron thick mouse brain tissue. And the other concern is whether there are methods to soften the tissue sufficient to allow isotropic expansion. And that will have to depend on specific sample that you're interested in using. So for today's talk, I will go through the detailed protocol for cell culture and mouse brain tissue. And after the talk, I'm happy to answer any questions. So just a review of the overall protocol of the 20EXM. You will start with a fixed cell culture or tissue. And the first step, we will treat it with NHS chemistry to anchor the proteins. And we will do generation of this tissue to embed it in the hydrogel. Then we do a step called softening, which is a step that disrupts interaction between proteins so that the tissue can start to expand. And we'll put the tissue in PBS. And at this step, we already expect the tissue to expand around 4-fold to 6-fold. And that creates space around the targets for the next round of antibody staining to occur. And finally, you expand in water to reach full expansion, and then you can perform imaging on a microscope, such as a spinning disc confocal. So before you start the protocol, there are two things that you should prepare in advance. One thing is we use hydrophobic glass slides and cover slips for the gelation chamber. And this is very critical because the gel that we form is actually very sticky and it's mechanically robust. So if the glass is normal glass slide, it will be very hard to take the glass slide off and retrieve the gel like in an intact way. So you will want to make the hydrophobic glass slides and cover slip in advance. And then the first 4 steps should be performed in a chemical hood with proper personal protection equipment because this involves organic solvent and hazardous chemicals. Essentially, we add hydrophobic silane, dissolve it in hexane, and you should do this in a petri dish. And you can put the glass slide, immerse your glass slide or cover slip in the solution for 90 seconds for the reaction to occur. And then you just need to take out the glass slide and cover slip from the solution with a tweezer, wash it with 70% isopropanol or ethanol first, and then water, and just allow it to dry in a 37-degree incubator. I usually keep the door open so the drying is faster. And you want to ensure that the glass is completely dry before you start to wipe off the residual reactants on the surface. And this is expected. Usually you will have some white residual chemicals. You essentially just sandpaper it with the dry Kim wipe, and essentially the glass slides will be ready. And you can test this easily by running water on the glass slide, and the water shouldn't attach to the glass anymore since it's been modified by the hydrophobic silane. And you also want to set up the glove bag in advance. So this is a sample glove bag that we have in our lab. You want to set up the glove bag on a bench that's near a compressed nitrogen cylinder or another nitrogen source. You want to connect the glove bag to the nitrogen source with a tube and use tape to wrap the connection very tightly to ensure it's airtight. And you can test if it's airtight by inflating the bag and just leave it there for a bit to see if it deflates. And if it's not airtight, you should see if you can fix it. If not, you can supply a small flow of nitrogen to keep it inflated. Although that will mean that you will run out of nitrogen more quickly. So once these are set up, we're ready to actually perform the experiment on the biological sample. And I will go through the cell culture first. So the types of cultured cells we've tested in our lab has included pretty commonly used cancer immortalized cell lines such as HeLa, HEC, and U2OS. We have also tested stem cell lines and induced pluripotent stem cells as well as differentiated cells. And we also have tested primary neuron cultures such as from mouse brain. And typically when we grow cells in our lab for expansion, we use one of these two setups. So one of them is a 16-well removal chamber cover glass, and the wells are quite small. Essentially, it has 4 components, and there's a tool you can use to cut the top part off. And then you can peel off the rubber, and then you can have the cells that's just on the glass slide. Alternatively, we also grow cells in 24 well plate with a cover slip at the bottom. So this is image of a single well, and we add a round cover slip at the bottom and grow cells on top of the cover slip. And when you're ready for expansion, you can simply take out the cover slip and perform expansion on top of the glass cover slip. And one note is that I recommend using thicker cover slips, such as number 2, and that makes the downstream steps easier, which I'll explain later. And finally, with coating, we typically doesn't use any coating, or we use protein-based coating, such as vitronectin for stem cells or polylysine. I don't think people have tested using metrogel coating. That might require additional optimization because they contain a lot of carbohydrates, and there's also other proteins, which might pose difficulty for the softening step, depending like how thick and the type of metrogel that you use. So this is how we grow the cells. And once the cell is ready, what we do is we will fix the cell either with 4% PFA or 3% PFA and 0.1% glutaraldehyde, depending on the type of target that you would like to see. For example, for microtubule and mitochondria, we observe that it's really critical to use the glutaraldehyde fixation to preserve the structures. Whereas if like the structure is not sensitive to degradation, 4% PFA should be enough. And after fixation, we will perform immunostaining. You can find the protocol. So all of the protocol that I will cover is also published with the paper in supplementary note 1. So you can access that online for free. So you can perform primary and secondary antibody staining on the cell culture. And this can be fluorescent or non-fluorescent because we will use a tertiary fluorescent antibody to boost the signal after the softening step. And finally, you want to treat the cells to anchor the proteins. And how this works is typically we use one of these NHS chemistry-based anchoring reagents. What it does is essentially it reacts with the proteins inside the cell through the NHS moiety and install a acrylate or acrylamide moiety onto the protein so that that can be directly incorporated into the polymer network. And in the original ProEXM paper in 2016, we reported using ACX. And in this paper, we have switched to only use AX because we haven't noticed the performance difference between the two, and AX is like significantly cheaper than ACX. So that can save some money. And we have also reported another anchoring called the GMA. That's a epoxide-based anchoring, and that should be also compatible with 20XM, although we haven't tested that in our lab to fully publish an optimized protocol yet. All right, so after these steps, you're ready to start expansion of these cell cultures. And if you're using the 16-chamber well plate, so now you can use this cover glass removal tool. Essentially, it's a device where you can slide the chamber into, and there's a wire that will cut through these 2 layers of rubber. And you should be able to get the bottom part of the rubber that's still attached to the cover slip, as shown here. So you remove the top part and you have the black rubber with cells in between, and then it's covered in PBS because you don't want the cell to dry out. And what you want to do is use a tweezer to carefully peel the rubber off this cover slip, and that could take some practice. It might be nice to practice without with like a chamber that doesn't have cell in it first. And the trick is to not, when you peel off the rubber, don't pull straight up because that will create tension that will break the glass. You want to peel it horizontally to kind of like peeling like a page of a book kind of motion. So once you remove the rubber, you will have a cover glass that has cell on top covered in PBS. And the way we construct the gelation chamber is now we take one of the hydrophobic glass slide that you made before, and we wrap parafilm on top of it to create these little spacers that will hold the cover glass up. And the parafilm is around 170 micron thick. So that's a pretty good thickness for the gel that you will make. And you just need to put the cover glass on top of the parafilm strips, as this is the cross section, and that will be ready for you to move it into the glove bag. And later on in the glove bag, all you need to do is to replace the PBS with the gelation solution and flip this cover slip upside down to finish the construction of the gelation chamber. If you are using the 24 well plate, this could also be a little challenging to take the cover slip out of a 24 well. The trick that our lab use is to use a needle with a bent tip. So that's kind of like a hook, and you can use the hook to lift up the cover slip and use the tweezer to remove that from the well. And similarly, once you remove the cover slip, you can construct the parafilm strips and place the cover glass on top with the cell facing up and PBS covering it. So once you've constructed the gelation chamber, you can now place the gelation chamber in the airtight container. And here we usually just use like a airtight lunchbox that's exclusive for the lab. So you use the lunchbox and you put a damp paper towel at the bottom. And I usually use the lid of the 24 well plate as the platform on top of the damp towel and place your gelation chamber on the hydrophobic glass slides on top. And the platform, like the platelet, can also be used as a platform to hold these slides up when you are in the glove bag, which is quite helpful because it's really hard to hold on to these slides directly with the gloves on. So now that these are constructed, you're ready to make the gelation solution. This is directly copied from the supplementary note 1. Here, you just need to dissolve the sodium acrylate in acidified tris buffer. I usually make the acidic tris buffer in bulk in advance. And that's 10% 1 molar tris HCl pH 8, 20% 1.2 molar HCl in water. So it's pretty acidic, and that will speed up speed up dissolving the sodium acrylate, and also will slow down the gelation later on. And then you want to dilute TMAT tenfold and add 7.5 microliter of the diluted TMAT into the sodium acrylate solution in the glass vial. And then you want to add DMAA. One note about DMAA is that DMAA is very viscous. So for this step, you want to measure the accurate volume, which I use the pre-wetting technique. So you use a pipette to draw up DMAA first, and you want to push it down to the first level, like the normal level, and then draw up again. So that will pre-wet the pipette and ensure that you have enough DMA volume. And when you add it to the glass vial, you just go down back to the first level again. Like don't go to the second level. And then your vortex. And at this step is when you will find out whether your gelation solution is good, because the solution should be colorless and non-cloudy. If you see any precipitates or is slightly yellow or there's any oily substance in it, that means the quality of the sodium acrylate and DMA is not good. And using that gelation solution could result in like faster gelation than expected or lower expansion factor where the gel couldn't form. So we put all the catalog numbers and vendors for the chemicals we use in the protocol, and I would highly recommend using the exact chemicals that we use. And if there's any problem with the chemicals, we would be happy to answer any questions. And finally, you want to make the initiator solution. So you dissolve potassium persulfate in water, and you want to vortex for 2 minutes because it takes some time to fully dissolve. And you don't want to put this solution on ice ever, because once it's on ice, it will precipitate out again, and you need to wait for it to warm up and dissolve back again. So once you've made the gelation solution and the initiator solution, you can put stuff into the glove bag. So I usually have pipette tips, the airtight humidified chamber that you just constructed with the generation chamber within. I would put the initiator, two empty vials, hydrophobic cover slips, transfer pipettes and tweezers in a box, and I will put the pipettes in. And one trick is to put your pipette tip on the pipette before you put them in the glove bag, and also to set the volume to what you want to use later, because that reduces the amount of things you need to do in the glove bag. And the glove bag is not easy to use. There are easier ones, but they're more expensive. So you can decide if you want to buy fancy ones. But after some practice, and these people in our lab, they are pretty adept at doing this. So once all the glove bag stuff is set up, you're ready to remove the oxygen from the generation solution. So you want to do this in a chemical hood and connect your gas dispersion bubbler to a nitrogen source. And the nitrogen flow actually needs to be minimal, like a minimal flow, just you can barely hear the sound when you hold it next to your ear is sufficient. You don't want more than that because that will cause evaporation. The generation solution might freeze. Then you probably need to make the generation solution again because then the water content changes and things might precipitate out. So if you're using this for the first time, please consider practicing it with just a vial and water so you can kind of like get a sense of how strong the nitrogen flow should be. And the other thing is, if you look at the video, when I put the glass bubbler inside the solution, I will twist it to make sure that it's fully wetted. Because if it's not fully wetted, the bubble wouldn't come out as vigorously as this, and then the removal wouldn't be complete. So all you need to do is to keep the solution on ice with the bubble going for 50 seconds, and that's enough to remove the oxygen from the solution. And then you want to cap the valve quickly after removing the gas dispersion tube to minimize oxygen exposure. And once this is done, you should just move the glass valve into the glove bag, and you should be ready to start the final steps of generation. So now everything's in the glove bag, and what you want to do is you want to turn on the nitrogen flow, and this will start to inflate. You want to use your hand to hold onto the seal for it to inflate to almost 90%. And then you want to purge the glove bag by pushing down on the top to push most of the gas out, and do this 3 times. So you're purging the bag to ensure that it's mostly nitrogen in the bag by the time you start performing the experiment. And you also want to make sure that the airtight chamber is not capped, because then nitrogen wouldn't be purged inside the chamber. So once you've purged it 3 times, you can seal the glove bag and it will fill up naturally. And then you can mix 411 microliter of generation solution with 20 microliter initiator solution in an empty centrifuge tube. And I would just flip the tube upside down 5 times to fully mix the solution. And then you use the transfer pipette to remove the PBS from the from the cell culture and add 50 microliter or more, depends on the well size. I will use 50 microliter for the 16 chambered glass and use 100 microliter for like 13 millimeter round cover glass to basically cover the cell, and then use the tweezer to flip the cover slip on top of the parafilm strip to construct the generation chamber. So that's from step E to step F. Basically, you replace the PBS with generation solution, and you flip it upside down. And now the cells are facing down in the generation solution. And now you just put this inside the airtight humidified chamber, cap the chamber, and then you can take it out of the glove bag. And you want to keep it in the dark at room temperature for 2 hours for the gel to form. After 2 hours, you can use a razor blade to pry off and flip the cell culture cover slip. And this is where number 2 cover slip really helps, because thinner ones might snap at this step and thicker ones is much easier. So once you flip it over, you want to cut this gel into a right tetrazoid shape. And this is really important for cell culture because you want to make sure that you know that the cell is facing down. Because if the gel is flipped and the cell is facing up, you will never see it when you image. So cutting it into a red trapezoid shape allow you to quickly tell whether the cell is facing down, which is the right direction. So after cutting, I just put it in a 6-well plate and add lysytrypsin protease and let it incubate at 37°C overnight. And after 37°C incubation, you can wash with PBS 2 times for 15 minutes to remove the protease. And now the final step is you want to do immunostaining to boost the signal. So we're talking about like a 8,000-fold spatial volume expansion here. So the signal gets very diluted and some signal amplification is needed. And in our experience, like how we do this is we just apply a tertiary antibody. If you can't find a tertiary antibody, a secondary antibody is possible, but the signal won't be as strong. So to stain tertiary antibody, we basically use a staining solution based using normal donkey serum and Triton. So you want to block the gel with blocking solution for 2 hours at room temperature. We usually use 200 microliter, but you're free to add more if the gel is large. You want to make sure the gel is fully immersed. And then you want to aspirate the blocking buffer and add in the staining solution, which contains the boost antibody. And we usually use 1 to 200 dilution of the standard secondary or tertiary antibody solutions. We have more details in the supplementary table 4. And then we leave this in a 4C fridge overnight for the antibody to diffuse in. And the next day, you just need to wash this with the washing solution, which is basically Triton in PBS, for 30 minutes, 3 times, or 4 times to wash out the antibodies. Finally, when you're ready to expand an image, so the gel, if you cut 0.5 centimeter after expansion, it will expand 20-fold. So that will be around like 10 centimeters. So it's quite big. Am I doing math? Yeah, 10 centimeters. So it's very big. So we recommend using an imaging plate. So we also have the catalog number in the protocol. It's essentially like a plate, but it's just like one huge plate with 1.5 glasses for the microscope. And that's pretty important because the 4-well plate will not be able to fit it. Or you can cut and then do it in a different well. That's also feasible. So you want to make sure that the right trapezoid shape is correct, that the cell is facing down. And then you just need to add water and wait for 20 minutes without shaking, like it's not necessary, and repeat this 2 to 4 times or until the gel no longer expands. And then typically we just remove water around the gel with transfer pipettes and Kim wipe and transfer to the microscope for imaging. And you might, depends on the gel shape and size. Sometimes you will have like drifting on the microscope. And if you look at the 2018 paper, I think we talked about like you can try to coat the imaging plate with polyl lysine or silane. There's different methods to like kind of like covalently attach the gel to the glass, and that will solve the drifting issue. But then you won't be able to take the gel off again, so you have to decide what's best for you. All right, so that's the cell culture version of the protocol. And next I want to talk about tissue. So in the study, we reported mouse brain tissue and spleen and kidney. The main difference is that the brain tissue, we support post-expansion staining, which means we simply expand the brain tissue, and then we perform primary and secondary antibody staining only after the softening step. Whereas for spleen and kidney, the softening step we use for brain is not sufficient for spleen or kidney. So we'll have to use the proteinase softening step. And because of that, we recommend doing primary, secondary antibody staining beforehand if you are working with more tough tissues such as spleen and kidney. But other than that, it's very similar thickness, fixation method, anchoring method. So with the brain tissue, once you perfuse it with 4% PFA, slice it into 50 micron. We've also done 100 micron if you want slightly thicker tissue, but we haven't tested tissue thicker than that. Treat it with AX, and then we do micro dissection because it will expand a lot. So we typically micro dissect mouse brain slice into pieces that's less than 2 millimeter by 2 millimeter. So it's quite a small piece, as shown here. So you can see this little piece floating in a droplet of PBS. And so after this is gelled, you will still need to use the imaging plate because it expands a lot. So essentially, it's a similar idea of constructing the gelation chamber, but slightly easier. So you just need a hydrophobic glass slide with parafilm strips, and you put 2 drops of PBS on top, and you just want to put the microdissected tissue inside the droplet. And when you're inside the glove bag, you just need to remove the PBS without removing the tissue, and then add in gelation solution and flip it. Sorry, not flip, and place the cover slip on top to finish the construction of the gelation chamber. And similarly, we put the gelation chamber into the airtight chamber in a similar construct, and all of the steps following are going to be similar. So now you want to make the gelation solution, which is also the same as I described in the cell culture protocol, construct the glove bag, and that's also going to be similar, and remove the nitrogen, which is also similar to cell culture protocol. And here is where it differs a little bit. So when you are in the glove bag after purging, you want to add 4 microliter of initial solution to 411 microliter of gelation solution instead of 20. And that slows down the gelation so that the gelation solution can diffuse into the brain tissue. And what you want to do is you want to remove the PBS and add the gelation solution to the brain tissue and incubate it in the gelation solution for 15 minutes. And it's important you have a damp towel in the chamber and you want to cap it during that 15 minutes because you don't want the drop of gelation solution to evaporate while you're waiting for the incubation. So after 15 minutes, then you use a tweezer to place a hydrophobic glass cover slip on top to cap the gelation chamber, and now it's complete. So now you can take it out. And with brain tissue, we keep it in the dark at room temperature overnight. So that's 16 to 20 hours for the gelation to complete. So this is from step B to step C. So afterwards, it's very similar. You want to cut out the brain tissue. And for brain tissue, it's not that important to cut in the trapezoid shape because it's thick enough, you'll probably be able to see it. And for digestion, we use this denaturation buffer. Essentially, it's SDS with DTT. And we heat it up at 95 degrees for an hour. That will denature the proteins and soften the gel, and then that will be able to expand. So we wash it with PBS twice for 15 minutes. And then now it's time to perform the staining. So the staining protocol is actually basically identical to the cell culture protocol, but instead of using a tertiary antibody to boost the signal, since you didn't do pre-expansion immunostaining, now's the time to perform primary and secondary antibody staining. So with that, you can basically do this twice to stain primary and secondary antibody, and then expansion is very similar. You want to fully expand this in the imaging plate, remove the excess water, and move the microscope for imaging. So with that, I want to thank Ed and Laura for supporting my PhD. It's great to work on useful technologies that I hope a lot of people benefit around the world. And I want to thank especially Tay and Harley who worked on this project with me, as well as Ryan from Peng Yin's lab. And just a big thanks to our collaborators in the lab and around the world, and also on all the funding sources. And I'll be happy to take any questions.
Excellent. Thank you so much. This was a great talk. And we have quite a few questions. So I'm going to start out with a question from Thomas. Is Halo tag labeling compatible with this protocol or other EXMs?
Yeah, so we have tested different tags, and I'm not sure about halo tag. So the main concern, so more information will be useful is whether you're doing cell culture or tissue and what softening method you're using. Because if the halo tag is somehow degraded during the softening step, then it wouldn't work. The safe bet is it will work if you perform the halo tag before expansion. So whatever you want to visualize, want to like click on the fluorophore, you can try to do that before expansion and then expand. And if it would be great if you can further amplify the signal to make sure you can see it after expansion. Yeah.
Cool, thank you. How good are these gels if you want to use a vibratome sectioning on them with tissue inside?
Yeah, so we haven't tested Vibratome. I think other expansion protocols have reported using cryostat to section the gel. And I would recommend looking at those studies. My experience is usually these gels are not really thick. So when we expand cell is definitely not a problem. When we expand thin tissue slices with confocal, you can almost, especially if you use 10x objective, then you can almost image throughout the tissue. So I think it really depends on what the tissue thickness you need to do to see if Vibratome is necessary.
Cool, thanks. I have another question by Thomas about staining. What about DNA staining like fish? Is it possible to combine it with IF and then do the expansion?
Yeah, so DNA staining is tricky with expansion. So it's actually a long-standing question in the lab because DNA themselves are not directly anchored to the gel. And DNA, they're long fiber structures. So when you expand, they might slide and move around. So if you look at, I think there's a recent DNA expansion paper from the Bolsonaro lab at Harvard. They actually have to generate a lot of the probes before expansion to circumvent this problem. But like in terms of labeling, I think it's definitely possible, but the difficulty will be to determine whether the DNA ultrastructure is preserved in the process of expansion.
Thank you. Valentine asks, have you evaluated different initiation methods such as thermal or photo initiations, and could those potentially alleviate a problem of sample equilibrium or equilibration, with gel prior to gelation?
Yeah, so in the beginning we looked at different like initiation methods. Um, and then we ended up just choosing potassium persulfate because that's what most expansion labs are already using. I think other methods are definitely possible, but you will need to do optimization because the polymer kinetics might be different.
Thanks. Valentine also asks, or Valentine, have you checked if potassium is critical versus traditional APS?
We haven't. We use potassium because that's what the original chemistry paper used, and that's what X10 paper used. And like since it's not like super expensive or anything, we just kept using potassium persulfate. Yeah.
There are a couple of questions transferring it into different tissues. So is it possible to observe collagen architectural patterns in human tissue samples?
So we haven't tested this gel with human tissues extensively. It's certainly possible, but with human tissue, I would recommend referring to I think there's a decrowding expansion pathology paper that we previously did. I think it depends on which type, like how the tissue is preserved, the tissue quality, and a lot of other factors. But definitely happy to chat more if you want to optimize that. Yeah.
Cool, thanks. Can you use this method for nanoscale synapse level connectomics and neurite tracing?
Yeah, I think it's definitely possible since we can visualize nanostructure of synapse and we can visualize the neural processes. We haven't performed any tracing with this technology in our lab. Yeah.
What confluence or cell density would you recommend? And that probably depends on what cells you're looking at, but
Yeah, I think it depends on the cell type, but typically I would use cell culture that's around 30 to 50% confluency. Yeah.
okay. Roger asks, what happens to cellular membranes? Don't they break down upon expansion?
Yes. So because of the softening step, like a lot, like the protein interaction will be disrupted, protein structure will be disturbed, and lipid, because of SDS, will be disrupted. But because the proteins are anchored to the polymer network, the spatial information should be preserved.
Thank you. Valentine asks, have you tried replacing DTT with TCEP? TCEP, sorry.
Yeah,
TCEP,
we
sorry.
we haven't. I think it's definitely possible. And then if you're having trouble with softening, I think there's definitely a lot of things that you can try to help with the reducing agent or like higher temperature or things like that.
Following up with that, Don asks, does DTT SDS softening also works for cultured cells?
It depends. So we actually observe some problem expanding of cells fixed with glutaraldehyde and softened with SDS DTT. So I think it really depends on the fixation of the cell, the cell type. Like what you want to prevent is that if, say, the cell is cross-linked because of glutaraldehyde or other things, you might need to use protease to actually cut those cross-links for the gel to fully expand.
How's the physical property, the sliminess or rigidness for the DMAA gel compared to normal or more traditional EXM gels?
It should be quite easy to handle, especially when it's in PBS. So we don't recommend handling when it's in water, when it's fully expanded, because then it's so big and it's more fragile. It's really difficult to handle. So we recommend all the handling step to happen in PBS. And usually in PBS, even if you just pick the gel up and transfer it, it shouldn't fall apart because it's quite robust. Yeah.
Wait, there was a question. There's so many questions. I'm catching up. Pooja, I hope I pronounced that right, asks, does this technique focuses only on one protein at a time, or can you look at multiple proteins or, for example, interacting proteins as well? And
yeah, you can definitely look at multiple proteins. So for the synapse example, we use 2 proteins. And actually, like that experiment is actually 3 because that's in the Thy1 YFP mouse brain tissues where we're actually also seeing YFP. So it really is like compatible with just normal fluorescence imaging. So like depends on how many channels your microscope have, you will be able to use like multiple proteins. Yeah.
Roger asks, what about labeling membrane-anchored proteins? If the membrane is being dissolved, does it expand?
Yeah, that's a good question. So we haven't tested the preservation of membrane-bound proteins, but the hope is like they will be modified with NHS chemistry, and then some part of the protein will be captured by the gel. Yeah. But the membrane won't be there.
Yes. Sarah asks, asks and says, wonderful talk. Thank you so much. Would it be possible to visualize small membrane lipids such as PIs?
Lipid, we recently, our lab actually have a lipid expansion paper, but I think that one used the lipid tag to visualize the lipid. To directly anchor lipid, you will need to optimize, for example, using an anchor that can react with certain lipid moieties to anchor them to the gel, but we haven't really explored in that direction. Yeah.
Thanks. Valentine asks, how did you come up with the idea of acidification of the gelling solution?
So the problem for that was that with tissue, you don't want the gel to gel too fast because then it won't have the time to diffuse in. And typically people use 4-HT for that purpose. So it's like inhibitor of radicals. But I looked at the kind of like the activation mechanism of the potassium persulfate, and then you can see it's a pH dependent process. So if you neutralize the pH, the activation of the potassium persulfate will be slowed down. And that turns out like it's helpful. It makes it much easier to dissolve the sodium acrylate and it also slows down the gelation.
Thanks. There's a question. Oops. Oh boy. How about the limit of the thickness of, for example, brain slices for a DMAA gel? What would you consider being the upper limit? 100 micrometers or 200 in your experience?
Yeah, so we have tested up to 100 micrometers, but we haven't tested thicker tissue than that. And the main limit is to see if it's possible for the diffusion to fully go through the center of the tissue. And then for that, you might have to optimize the gelation solution further to slow down the gelation.
Carmen asks, how does that method compare to LeCon, the 16x expansion paper that was published recently?
Yeah, so we haven't performed the side-by-side comparison of the two methods, but LICON is an iterative expansion protocol. So it just depends on what you prefer. I think like in terms of workload, there's definitely less workload for this protocol. But we haven't really compared, say, like the preservation of ultrastructure of the two methods.
Thanks. Has anyone tried this in plants, cells, or tissues?
I don't know if anyone has tried this with plant cells. That's a great question. I would say if you want to try this on plant cells, I'm happy to talk more. It will depend on like maybe look at if people have used expansion microscopy with plants and what modification they did, and then we can like think about what modification we can make to this protocol.
I presume it might require digesting the cell walls.
Yeah.
There's more questions regarding different organisms. Have you or anyone tried bacterial cells or protists?
Yeah, we have tried some bacteria cells. I think with bacterial cells, it doesn't seem like diffusion is a big problem. The big challenge is the softening because they make different cell walls, a lot of them are carbohydrates. You will have to think about how to fully digest the cell wall for the expansion to occur.
And protists?
We haven't tried that, but that might be easier. I'm not sure.
Okay, Dario asks, I wonder how it expands cellular structures that were supposed to be more or less rigid, and how it will work on cellular organisms with a more complex cellular membrane or cytoskeleton.
Yeah, I think, at least with our lab's experience, we haven't really observed differences between different organelles when we expand the cell. I think the key is to ensure that you're softening like sufficiently so that like some things don't expand more than the others.
Someone is asking a microscope question. How critical is it to use spinning disk confocal to image versus a wide field or, I guess, a scanning confocal scope?
I don't think it's that critical at all. Since the sample's already expanded, you can use whatever confocal you want to use. We use spinning disk because that's what we have, and it has pretty good performance in terms of like scanning large areas. But yeah, if you want to use other microscopes, it's definitely possible.
Cool. Has anyone tried to visualize centrioles and flagella or cilia?
I know that there have been groups using expansion to look at centrioles and flagellates, but we didn't do that for our paper.
Okay, someone is asking about a more 3D aspect. Have you noticed any curvation of the cells or the tissue when you view them in the XY or XZ or YZ plane when they would be expected to be flat? So basically, how does it affect the Z?
Yeah, so we looked at Z-axis expansion and distortion of tissue, and they seem to be very consistent. We don't see any like difference in terms of expansion factor for Z compared to the XY.
Someone asks something regarding your boosting. When you add your booster or tertiary antibody, does it become a big cluster that actually could reduce the resolution and precision?
So the advantage of expansion microscopy, especially when you stain after expansion, is that the linker error, so the size of the antibody, is almost negligible because the signal is already expanded and the antibody is like 10 nanometer divided by 20-fold expansion. So that's like a 0.5 nanometer linkage error. So we haven't really observed any like differences made by the boost antibody.
That almost answers the next question from the same person. Since the antibodies are already sizable, depending on what antibody, nanobody, or antibody fragment you're using, obviously, could that cause a problem if you want to visualize a complex that contains multiple proteins and you want to image all of them at the same time?
Yeah, I think, yeah, like I said, I think it's definitely it's great to use post-expansion staining, although that's currently only validated in mouse brain tissue.
Okay, there's 2 more questions regarding different organisms. Has anyone used it on organoids, and has anyone used viral particles that are between the nuclear membranes?
So yeah, so I'm not sure if anyone has used an organoids. I think I've heard about it. I think people might have. Viral particles, do you mean like expanding virus themselves? Because that's tetragel from our lab did, but not like it's like purified viruses, not within biological context.
Okay, the specific question is, how would you visualize viral particles between the nuclear membranes if they're localized there?
I'm not sure.
Might be a very too specific of a question. All right. Can your technique be combined with other super resolution techniques or labeling strategies to further enhance the imaging capabilities?
Yeah, I think it's definitely possible. And like several groups in the world have already tried to combine expansion with other super resolution, and this gel is just a gel. So I think those improvements are definitely possible. Yeah.
Okay, I think I apologize. There are so many questions. If I've missed one, I think there's one more on staining. Have you tried chromogenic staining in the expanded tissues?
We haven't, no.
Okay, I think this was great. This was a fantastic talk, and thank you so much also for this very active Q&A session. And well, maybe